Good morning, ladies and gentlemen, and welcome to the opening session of day three here in Hall A at our longevity conference. I hope everyone here has had a splendid time filled with engaging conversations over the last 2 days. So, it is my privilege to introduce today's chair who will guide us through this engaging session. We have Dr. Smita Hegde, a distinguished consultant ENT head and neck oncology surgeon from HCG Global. Please join me in welcoming Dr. Smita Hegde onto the stage.
A very warm good morning. We've had 2 very exciting days. We've spent a lot of time talking about longevity, about health span, about lifespan. And all of this information to us has come from the large number of clinical trials and cohort studies that are happening across the globe, especially very exciting research that's coming out of India as well. Today, we have very prominent four speakers who have done some amazing work in the field of aging and longevity. And I think it's great that we are here at such a juncture where we are now able to identify biomarkers for aging. We are able to identify biomarkers for frailty. We are actually able to quantify how somebody is aging and how soon they will age. Probably not how long, but there's still hope. So without much ado, our first speaker for today is Prof. Arvind Ramanathan, who's the principal investigator at the Institute of Stem Cell Science and Regenerative Medicine, InStem, Bengaluru. Please.
Okay, it's really a privilege to be here. I want to thank the organizers, Deepak, of course, and many of his amazing lab to have put this really amazing group of people together. So, without any delay, let me just tell you that we have had these amazing 2 days where we've heard about so many of these clinical outcomes and basic sciences and all of these things. In the end, we need to integrate all of this in human studies. And I really think the main challenge here is going to be how do you take these insights from the bedside that all our wonderful clinicians and all the insights that we get, and how do we bridge this science gap between basic scientists, biochemists, and molecular biologists, and see what kind of interventions one could and how to even think about this. So I'll just give you an example of, I think this is really interesting and important study that I've been performing. But before that, I'll just say I'm from the Institute for Stem Cell Science and Regenerative Medicine, not very far from here. It's a Department of Biotechnology Institute, which focuses on regeneration, and my lab specifically focuses on skeletal muscle homeostasis and aging. We do multiple things in the lab. I'm not going to go into it. We're very interested in cold shock and senescence. But today, what I'm going to tell you is the ability to bridge basic sciences with the wonderful study that I'm going to tell you about with Baptist Hospital and Via Vikas and others, where we have gotten some interesting insights into how people age and how we can use these circulating biomarkers and even take it a step further and see how we could learn from these to move them into interventions and the kind of progress we are making in the context of organoids and things to see how we can move. I'll tell you the story. Uh, so first of all, why should we even focus on skeletal muscle, right? And this is something very empirical that you all know from our own lives, but a graph like this makes it quite clear, right? After the age of 40, you, with some weight training and so on, you can slow this progress down, but it's inevitable that you lose a significant amount of muscle mass by the time you get to 65. So that is one major thing that we know, that we have experienced, and that we know from our family members. So what is sarcopenia? On the whole, sarcopenia literally means loss of skeletal muscle, but there are multiple components to it, right? Once you don't have skeletal muscle, we realize it is the engine in the body. This is the sink of glucose and all your work, heat generation, metabolism. When you lose the skeletal muscle mass and its function, the metabolic impacts are tremendous. You can try to go fix type diet, or, no, the glucose homeostasis issues, but it comes directly from the loss of skeletal muscle mass. Similarly, we know it does the work of moving, the work of life itself. So clearly, you'll have issues like frailty and so on. The other remarkable thing about skeletal muscle that we know now is like a real supporter of memory and cognition. There are these repeated studies which tell you that one of the best ways to improve your memory is to exercise regularly. And we in the lab, and I'm going to go into it, are really uncovering molecular factors that the muscle is secreting that directly changes the homeostasis of of the brain. But we'll not go into that. But two other factors I just want to say. This is well known. Here is an interesting study. If you want to predict the longevity of older adults after the age of 55, there is a very nice study that was done by the National Health and Nutrition Examination Survey, where they were just measuring skeletal muscle mass, and mass index was measured by kgs per meter square, and here are different quartiles. It really told you that just by measuring skeletal muscle mass, you could predict how long somebody was going to live, and you can tell that so essentially. From the fourth quartile to the first quartile, you can see a major effect that skeletal muscle at least correlates in terms of survival. That already tells you that this is a very, very clear factor. The other thing that was came out a few years ago was that they used these 460 400 more than 400,000 individuals from the UK Biobank, and they just measured multiple aspects of frailty and physical function and markers and all that. Remarkably, the hand grip strength was a major predictor of all cause mortality. So this tells us more and more that we know these correlations are happening, but this tissue is playing a remarkable role in how we age, and probably taking care of this tissue might be sufficient in some sense, because it is our body is such a homeostatic engine that everybody talks to everybody, that this might be a major target for healthy muscle. So I'm going to tell you a little bit about what I'm going to tell you about today. This has been an ambitious study that we have been working with a number of people in this audience, including Baptist Hospital and others. I'm going to tell you a little bit about. The idea is mapping. So the thing is, skeletal muscle is the second largest tissue in the body after skin, and it secretes a lot of your metabolites and other things. And of course, we're not saying the circulating markers are just from muscle, but muscle plays an important role in the circulating markers. So can you, in a systematic, comprehensive way, map these circulating markers and learn something about, correlate them with clinical outcomes and others, and find markers which will tell you something about skeletal muscle outcomes, frailty, and so on. But taking it a step further, what we really want to do is see what kind of biochemical insights it's giving you. Would it give you insights on what pathways to target? And then I'm going to take you to a very, I think, critical aspect of science today, which is developing human models of aging and other tissues, that now we can use human skeletal muscle organoids to start testing and interrogating whether these can have an effect. So I'm going to tell you, just allude to a few things in this talk. The first part of my talk, before I get to the organoids, is essentially a clinical mapping of intrinsic and extrinsic factors that affect skeletal muscle aging. Right, and this, I think, was a remarkable pilot study that will lead to bigger and better things, that was done in collaboration with a number of people here, including I put AHPI, but who was a major driver of this study. Right, so the idea is, okay, let us take a clinical cohort, look at their clinical outcomes in terms of skeletal muscle, and see what we find. Right, some of these things you just have to take a leap. So we did a pilot study. We didn't know what we were going to find. So this was, we call it the Bangalore Sarcopenia study. It is in submission right now. Hopefully it will be published in the near future. But this is essentially the cohort that we had. Right, so we tried to have an equal number of male and females, and we made sure that they didn't have any, as much as possible during age chronic outcomes, but these were the profiles. And essentially what we did was a Sarkov score to decide whether somebody was sarcopenic or not. 0 and above was essentially sarcopenic in our study, and then we wanted to have it correlate to biochemical readouts. And as you can tell, in the most of the females in our study, and maybe this is a reflection of our society, are more subject to sarcopenia in some sense, at least based on and of course in the males we had at least half of them who were non-sarcopenic. So first question, how do you measure sarcopenia? That, I think, is an open question, but I think we have really come to fruition here. You can use imaging studies, you can use all kinds of functional readouts, and if you use a Sarkov score to know whether somebody is sarcopenic or not, what would be the rapid study one does to distinguish? So this is really nice work done by Dr. Elizabeth George at Baptist Hospital and the orthopedic team there, where they did this amazing, these clinical studies are only as good as clinicians are. They have such a keen eye and a sensitivity for these kinds of measurements that biochemists like us can just come in and run some samples, but all of that depends on this really keen study. But I think one specific outcome I just like you to, this is just essentially a statistical study which tells you how sensitive and specific are any of these readouts to predict sarcopenia. And we find very nicely that the dominant hand grip strength is a really, really good measure and determinant of sarcopenia, which tells us now going forward as we increase populations and better studies, we have zoned in on one study, one measurement that will tell us very clearly. Okay. Then it's always interesting to compare yourself to people have done dominant hand grip strengths across the world. It's interesting. This is our BSARC study. And you can tell this blue color is males. So anybody less than a grip strength of 27 in our study, what could be classified as sarcopenic, and with females it was 16. And it's interesting to compare it to the world. Very nice to see that number of studies have similar numbers around the world, giving you a little sanity check. Also interesting to see how people in Nordic age and maybe better in terms of skill strength, right? They're 35 and so on. So, but it's very interesting. So now we can take our study and map it across the world and start comparing to how are we doing compared to others. So, with lack of time, I'll just tell you that now we went and measured hundreds and hundreds of metabolites from blood. Okay, and this is the metabolite classes, right? Nucleotides, organic acids, amino acids, carbohydrates, number of dietary sources, lipids, and so on. And we just let's see what it tells us. Number one, I'll just tell you a few insights, and then we can always talk offline. One interesting thing that we found that the NLR ratio, which in the clinic is often a very, very good measure of inflammation, correlates very nicely with sarcopenic. The red color dots are sarcopenic, blue are non-sarcopenic. And arachidonic acid that we find in our LCMS, which is of course a precursor to a number of inflammatory lipids, are also increased. So good. So we can see that interestingly, there's a clear correlation with inflammation and a source of inflammatory lipids. And I just wanted to know that it is time that we went much deeper into understanding nutrition, metabolism, and all of these things. We know amino acids are good. People use these words, but we know we have to really get to the granularity. Now we are getting some interesting features about, so the blue colors are non-sarcopenic, red colors are sarcopenic, and you can see that there are a number of amino acids which behave differently, right? You have homoarginine, which is a very interesting natural source of nitric oxide. That we know. I mean, I have no evidence that is the mechanism, but it starts compelling these thoughts in your mind. A major biological source of nitric oxide, which is important for vascular health, is enriched in non-sarcopenic patients. Interesting. So things like that you start learning from these sorts of studies that you can now start going and testing. Similarly, I mean, we won't go into this, but the number of metabolites that we have are significantly decreased in sarcopenic and non-sarcopenic. And these might start giving us insights on pathways, inflammatory pathways, metabolic pathways that could be drivers of sarcopenia, if you will. So won't go too deeply into this, but in the end, we are living in the world of AI, machine learning, and all of these things. And so essentially, you ask the question, I have measured these hundreds of things. There are sarcopenic, non-sarcopenic people. You ask the AI that, what do I have to measure to tell if somebody is sarcopenic or not that I should focus on? So we did something called support vector machine analysis, and using all of these samples. Essentially, it's quite a simple algorithm in principle. Essentially, you're essentially drawing a hyperplane that's able to distinguish between sarcopenic and non-sarcopenic over multiple metabolites. And we find a very important panel of metabolites that seem to predict very clearly whether somebody is sarcopenic or not. And hopefully, next time we meet, I'll be able to tell you a little bit about a natural product that seems to be highly enriched in non-sarcopenic patients, which we really do think could be a possible intervention by some early studies in the lab. But those are the kind of insights one gets. The second part, as we all know, we have heard so much about microbiome and all of those things. I mean, life is complicated. There's metabolites, there's muscle, there's microbiome, and all of these are networked in some way. I will not say that we have a full understanding of it, but what studies like these provide are some major data sets like that we can work with multiple groups to get better insights. But why? So I just wanted to tell you that when we measure the gut microbiome of these patients, and we ask subjects, and we ask who's sarcopenic or not, are there taxa that might be able to help you distinguish? One of the interesting things you find is that when you measure the gut microbiome, just based on diversity and distance from sarcopenic and non-sarcopenic, it tells us, look, there is no one thing called a gut microbiome of sarcopenic versus not. It's actually, that's a little more granularity. So we'll need to go a bit deeper into it. But essentially to say that if you just look at the diversity of the microbiome, there are two types of microbiomes. There seems to be one group we calling G2, which is closer to your control, but yet sarcopenic, and there is one which deviates completely. And they also differ in terms of how they, you can say both of them, of course, can very clearly predict dominant hand grip strength, but they have other different features in terms of inflammation and so on. That now tells you, look, the microbiome composition is not as simple, and there are certain microbes possibly which might be contributing to inflammation and others. Now, how do we go dig that deeper? Is I told you we have this major metabolite data set. We have the major microbiome, we have the major metabolite data set that told us whether you're sarcopenic or not. And you have these large number of microbes that are either increased or decreased in sarcopenia. Can we now start building maps of whether who's talking to whom? Could we really try to understand? So, I mean, these are, we have just finished these studies, but it's very interesting that there are certain microbes that are positively correlated with certain metabolites, which is known that they can metabolize spermidine. And we know spermidine is associated with aging and inflammation. That now triangulates a few things for us. We don't have the causality yet, but now it helps to start thinking, okay, microbiome here, metabolites associated with aging here, sarcopenia here. Can we triangulate and really try to understand how life is working in some way? And it might give us insights into possible probiotic supplements and all of that in the future. Similarly, we see other things that are correlated. But on the whole, I just want to tell you that you can correlate the pro-inflammatory metabolites that we know from literature with a couple of taxa that we know have been associated with inflammation and with sarcopenia. And we'll really try to triangulate the relationship between systemic inflammation and gut dysbiosis. So that's where we're trying to get. So what we have finally in this study is that we have panels of metabolites which are able to predict sarcopenic or not, which are giving you metabolic insights and possible supplement clues as to what you might want to do. And lastly, trying to bring in the really complicated gut microbiome into this axis to give us a complete picture of sarcopenia. So now when you have all of these things, somebody in a lab, I just really need to know, where do we take this further? Yes, we'll increase the cohorts, we will do more analysis, more and more analysis. But it almost feels like if you have to do thousands and thousands to get the insight, can I take what I learned from these 40 and take it to the, some sort of a endpoint in terms of making lives of patients better? So I just wanted to underline a very important development that's occurring right now in the world that we really have to embrace right now is the making of organoids. The days of using animals to understand human biology, human aging are behind us. It's still very, very good to understand physiology and connections. But the fact is, just I don't want to go into the granularities, but our muscle is completely different than a mouse's muscle. Our muscles are full of different fiber types, more mitochondria, more glycolytic, fast twitch, slow twitch. The compositions across different animals, from small animals to large animals is completely different. So we really have to take into consideration how we learn about muscle loss, how it's different in a mouse versus a chimp versus humans. So ideally, we should go to humans very quickly. How do we do that? It's not been easy so far. And this is also another driver. We are working with Dr. Reddy Labs and others who are also worried that the FDA now has told you, it is still early days, it has told you that you no longer have to do animal tests to get a drug approved. And we are getting to the world very soon where AI and organoids and all of that are going to be the way by which we understand supplements and this. And so we have to embrace this. The writing's on the wall. The sooner we move to organoids and human models of disease and aging, the better it is. So our lab is taking a stab at it. I'm not going to go too far, but it's very interesting. Now, how do you model aging in a dish, right? So now we can take myoblasts from, so we actually have the approval and we are getting samples now from Baptist Hospital and Dr. Alex and others who are collaborating with us. We are getting primary myoblasts from young and old people. Now, what if you could build your own contractile muscle? So Ashwin and a number of people here in the lab, including Diksha, and you'll see those, are beginning to build these models where you can build these contractile muscle fibers that came from a specific human being. Now, you're just seeing a little post in a small 96 dish that is forming this. How much time do I have? 3 minutes, yeah. So how do you know it's muscle? So it's very nice. So you can do a lot of histological things that it's beginning to look a lot like skeletal muscle in terms of a smooth muscle actinin. You can see a number of markers of muscle, including myosin heavy chain, different fast twitch, slow twitch fibers. So it's beginning to build human muscle in the lab, possibly in a more personalized way. That now the combination of biomarkers, possible interventions, and organoids could be leading you towards a new age of trying to get it to the preclinical stage as quickly as possible. And we can do very, very interesting things. We have compounds that we're testing that improve muscle size. It's done in a human being, and we are testing a number of other readouts, like, we heard a lot about mitochondria and so on. Now here is a human muscle that has been treated with a supplement that increases mitochondrial function very reproducibly, very significantly, and you can measure many of these aspects of mitochondrial function. So now we are getting to the point where, good, you have supplements, interventions, you have human organoids, you can start testing supplements to see how they respond in terms of different aspects of muscle. So where are we going? We essentially, as we validate these markers and do more and more studies, we already have some interesting insights that can now be tested in these very novel, close to human preclinical of tissue. And hopefully the future is we can get towards a more precision nutrition, where these insights can be directly tested in human systems that can bring it closer to the clinic in terms of safety and efficacy. So I just want to tell, I've worked with an amazing group of people. Dr. Thomas and Dr. Caroline are already, I think I told you about them. There's a number of people who help us do mass spec and things I haven't talked to you about. There's an entire group that's working very, very hard, some in the audience, who actually are moving us into this preclinical age of organoids. So I just wanted to thank you and all these founders.
Thank you for that very exciting research that you shared with us. We have time for one question. The rest of you can probably talk to him during the break.
Very nice study, and I'm really looking forward to all your future studies. One thing is, we know that South Asians have worse muscle mass and muscle quality than the rest of the world. So, in your study, did you find the correlation between blood glucose levels, protein intake, and muscle mass?
So that's a great question. We did have in our questionnaire vegetarian versus non-vegetarian and so on. We actually, I didn't show you this, but there's this major difference between rural and urban. It seemed like our rural brothers and sisters are deeply affected by sarcopenia compared to the urban. And you can have a romantic notion of how beautiful less population, but it's all about nutrition, as exactly as you point out. Protein intake, quality of protein probably affects a lot about. So I think there are indicators to what you're saying, but that's a very, very important question. We don't have a clear answer.
Thank you.
Thank you, Dr. Ramanathan.